conduct action potentials at the same rate. And the two primary reasons that we talked about is that nerves aren't built the same. Okay, some nerves have what's called myelin, which we'll study in better detail. And others, obviously, they just have a larger diameter, which we made our conclusions over. But let's just review contiguous conduction. What type of cell type uses contiguous conduction? Are unmyelinated or non-myelinated cells. So our contiguous conducting cells are those cells that lack myelin. They work just fine, but they don't have this extra structure associated with them. Conversely, we learned that some cells do have something called myelin, and the type of conduction is called saltatory conduction. And what is the advantage of having myelin in a saltatory conducting cell? It's faster. We don't change in saltatory conduction. The events of an action potential don't change, but they just are conducted faster. Okay, so it's enhanced, if you will. So in other words, our voltage-gated sodium channels work the same, voltage-gated potassium channels, all that, the pump, the channels, all those things work the same, but because of just the physical changes that we'll see in the cell with the addition of myelin, causes the action potential to carry from point A to point B faster. Okay? So we learned that myelin enhances conduction, as well as we learned that larger diameter nerves conduct faster. Okay? Because we learned in lab why. What was the point of that? What are we changing? What about the cell is allowing the velocity to speed up? We've already talked about myelination, so we learned if a nerve cell gets larger in diameter, what is it about that large diameter that allows for this increased velocity? More surface area is one of the main reasons why. More surface area, more channels, and more opportunity for ions to flow across the cell membrane. And so what I want to conclude this chapter with is a few images from your textbook to compare and contrast a myelinated versus a non-myelinated cell. In contiguous conduction, we already kind of understood basically where one section of the cell, once it depolarizes, that section is then going to repolarize and hyperpolarize. And it's basically going to be in that refractory period. But the ions which it let in open up the gated channels in the next section of the membrane. Okay? So this compounds from one section of the membrane to another. So it progressively goes from the hillock down the axon and eventually to the axon terminal, which we'll see what happens there in chapter 8. Okay? So the big thing is, can you see how it just moves from one section to another? It's not like a flash point across the whole cell. It just progresses along the axon. That's called contiguous conduction. Okay. And that's summarized for you in figure 7.19. Okay, so we're going to make a reference to it. Figure 7.1 illustrates this contiguous conducting of action potentials along a non-myelinated cell. If we were to contrast that to cells that are myelinated, the myelin in this image is illustrated in the orange bands. It's oversimplified, but we can kind of get an idea where the nerve axon still operates like it would normally operate, like we saw in contiguous conduction, except for the space where action potentials are conducted are a little bit further apart. So it's described sometimes as like the action potential jumps from one spot to another. It looks like it's kind of hopping around, if you will. But it's still the same concept. Ions flow in, open up gated channels, and flow in and open up gated channels. So it's still the same concept, it's just more space between. Or in other words, we've said the axon is insulated. I have a picture in here that always gets hidden. I tend to like this little image as well. We can see the myelin in the blue. We can see the nerve axon in yellow. A lot of the images show the action potential like it's on the outside, but where is the action potential being measured? Inside, in the intracellular. Sometimes the images get confusing. But here in the far end of the image, we can see where the ions are flooding into the cell and then opening up gates in the next little node. The question is, hopefully naturally, the next question is, well, what is myelin and what forms it? Well, primarily it depends on where the cell type is. Dictates what cell type actually forms the myelin. And so the big difference is whether you're looking at the central nervous system or the peripheral nervous system. The cell types that form myelin in the brain and the spinal cord are called oligodendrocytes. And in the peripheral nervous system, the cells that form myelin are called Schwann cells. So in other words, the myelin's not another nerve. It's some other specialized cell type, an oligodendrocyte or a Schwann cell, depending if you're looking at a cranial nerve or a spinal nerve. And the image to the right kind of gives you an idea what happens. On the top image, we have a peripheral nerve in the light yellow, and you can see the Schwann cells in the yellow. Hopefully you can see the Schwann cells. I'm saying that you can, but I hope that you can see the Schwann cells in the yellow. And in the next panel below that, we can see very lightly the central nervous system nerve axon. But the oligodendrocytes are the kind of darker yellow structures. So they're not nerves, but they're there kind of helping out. And so we'll reface these when we actually get into the central nervous system. And so all it is, because it's another cell growing along the nerve axon, what happens is that Schwann or oligodendrocyte cell just kind of encirculates, encompasses, or wraps around the nerve axon. So basically we have phospholipid bilayer sandwiched upon phospholipid bilayer. It's just layers and layers of what? Lipids. Okay. And they're anchored down with proteins. Okay, so it's a big layer of fat, basically, there to insulate. And where there's a big layer of fat, we're not going to move ions quickly. So in other words, what's going to stay stable where there's myelin? The ions, and what can we extrapolate from that? The ions, and just the potentials in general. The cell's going to be where? At what potential? Probably at rest, right? Where it's insulated, it's pretty well going to be stabilized at resting membrane potential. Okay. And so anatomically, we've already learned that the third point down here, the purpose of the myelin is to increase the action potential conduction, or described here, nerve impulse transmission. But some of you guys noted as well in lab, it does give physical protection to nerves that are insulated. So this big layer of fat, right? So it physically gives it protection as well to the functional features that we talked about. So are all nerves myelinated? No. Figure 7.5 has some good images for you to illustrate how the myelin actually forms. We're looking at a cross-section of a nerve, just some nerve axon. We have the peripheral nervous system in yellow. And that Schwann cell in the peripheral nervous system, or the oligodendrocyte in the central nervous system, has to, remember we learned way back in chapter 1 or 2 how proteins can be used for cell-to-cell attachment. We can use proteins for cell-to-cell attachment, or adhesion really, between a Schwann cell and an axon. So the proteins aren't shown here, but it actually, the term is called anchors. The Schwann cell or the myelin-forming cell anchors to the axon of the nerve, and then it actually encirculates. And notice what happens to its cytoplasm. Notice how the cytoplasm keeps getting pushed or bulged out. And in the mature form, you can just see pretty well just membrane, adhered to membrane, just wrapped around. And a visual is, if you think about like a cardboard tube of a toilet paper roll, the membrane would be analogous to the paper, which is flat and pushed. And then if there was something extra cytoplasmic on your toilet paper, pushing out. So it's a live, living, metabolizing cell that's just wrapped around a nerve axon. Figure 7.5 contrasts the Schwann cells versus the arrangement of the oligodendrocytes. And so they've built a little bit differently. They're obviously in different locations. I want to show you a real image. This is a transmission electron microscope of a nerve axon that's been turned on in and cut, or what's called a cross section. So you're looking down into it. You can see the kind of cylinder-like cytoplasm shape of the cell. And if you look here in the cells forming myelin. If you look really closely, I don't know how it is projecting, but you can almost see, almost looks like the rings of a tree. Can y'all see that back there? And so that's just the membrane of the oligodendrocyte. Or actually, I guess it's a triaxillon cell in the peripheral nervous system. And so it just wraps around. And you can see how difficult it would be for some little ion to quickly diffuse through all those layers of fats. The next image that I have is just illustrating for you that the myelin here in the purple is actually anchored down to the axon nerve axon 2. Do you have any questions about the difference between a myelinated versus a non-myelinated cell? When we move on to chapter 8. Comfortable with it? Chapter 11? All right. Let's go to move to chapter 8. As I mentioned before, it's really going to complete the story with chapter 7 about how a cell, when it gets excited, and what happens. And so these two chapters run along with one another. Chapter 8 is going to deal with how a nerve cell releases its neurotransmitter and the effect which it has on another cell, whether it be, first we'll study another nerve cell or say a muscle or something like that. And so what happens is the nerve cell will form a synapse with some other cell, as I mentioned, whether it be a nerve cell or some effector cell like a gland, endocrine gland, exocrine gland, whatever, or some type of smooth muscle, skeletal muscle, or even cardiac muscle. We're looking at nerve-to-nerve communication. We can use some terminology. We can use the term presynaptic and postsynaptic nerve. And the space between the two would be called the cleft. The presynaptic cell, what do you think does? Sending the message called the what? Neurotransmitter. The postsynaptic cell is the cell that has the what? Receptors. So we could call it the postsynaptic cell, or we could call it the target, to be consistent with what we learned in chapter 5. But the space between the cleft is just that interstitial fluid-filled space in which the neurotransmitter diffuses across. We're going to focus predominantly on what's called chemical synapses, and really I've been addressing it in this fashion. But there are some synapses which are called electrical synapses, which we have an idea about when we became familiar with gap junctions. So I'll very briefly review or review an electrical synapse, but the rest of this presentation for the nervous system will be our chemical synapses, where an actual neurotransmitter is released between two cells. So our electrical synapse is just the way that action potentials can be conducted directly from one cell to another utilizing gap junctions. Right? So ions flow between cells through little gap junctions, and it conducts an action potential. And so where we'll see these are in our some types of smooth muscle, and certainly in our heart, and the cells that are all working together. I'm not throwing out the proper term at this point. There's some current research, kind of cutting-edge research, if you will, supporting that there are actually more electrical synapses than previously thought in the brain. And so, although we focus on the chemical or the neurotransmitter synapses, in the near future, I think that this thought is going to modify that, not just neurotransmitters, causing the brain to function the way that it does. Okay so far? Not the Mondays, I think. All right. So what we're going to do is look at two nerve cells and just had a nice image to summarize. I'll try to illustrate, I guess, what's going to go on. Here we can see a large cell body in this image. And if you look closely, you can see the terminal of one cell over in the left side. You know, moving to the right, you can see another terminal, and they're highlighted by little flashy signals, but it's just illustrating that cell-to-cell communication between a presynaptic and a postsynaptic cell. And we're going to look at the release of neurotransmitter into the space called the synaptic cleft. You have a similar image, figure 8.1. In your textbook, I like this one better, so I provided it for you in your handout. But the presynaptic cell does not have to synapse only at the dendrites. Okay? If the presynaptic cell does synapse at the dendrite, it's called an axodendritic synapse. Which color of presynaptic cell does this image illustrate an axodendritic synapse? The green one. Okay? And you'll notice the green cell, the rest of it's been cut off. You can just see what part of the green cell. The green cell is what? Not its dendrites. The green cell's terminals. Okay. That's the axon terminals. There's one reason I can tell is I can see some myelin. But the primary reason I can tell is my postsynaptic cell is what color? The yellow thing. And I can see the cell body. And so if some cell is communicating with it, it's got to be the presynaptic cell. And there's not a dendrite-to-dendrite communication that I know of. And so the presynaptic green cell is synapting on the postsynaptic cell's what? Dendrite. And we call that a what? Axodendritic synapse. Okay. Where else can a presynaptic nerve synapse at a postsynaptic cell? On the cell body, and what do you call that? Axosomatic. Very good. Axosomatic, soma meaning body. The axon terminal can synapse at the cell body of a postsynaptic cell. Anywhere else? At the hillock. It's called the axoaxonic synapse. What color cell is forming an axoaxonic synapse here in this image? The blue one. So the blue cell is releasing neurotransmitter to the yellow cell. The red cell is releasing neurotransmitter to the yellow cell. The green cell can also release neurotransmitter. So all of those collectively are called what type of cells wherever they synapse? We could call them each, those are different types of presynaptic cells. The yellow, hands down, is always in this case postsynaptic cell until we get to its terminal, and then it would release neurotransmitter and be called the presynaptic. So it depends on which synapse you're looking at. Looking at a presynaptic cell always releases neurotransmitter. The postsynaptic cell has the receptors. Your book also indicates that a synapse can happen at the terminal of a postsynaptic cell, and I don't know how true. I haven't seen enough evidence to support that. And so let's keep for this course up here, dendrite cell body, hillock region. Okay? Did I catch that? So we discussed those in the image. And the cleft is just the fluid-filled space between the pre and the postsynaptic cell. And because there's an anatomical space between, there's no direct ion flow from one cell to another. Okay? So there's not a direct, so presynaptic cell doesn't directly pass its action potential to the postsynaptic cell. Okay? There's going to convert this electrical event to the release of a chemical or a neurotransmitter. And it's definitely unidirectional. What does that mean? One direction. Pre always communicates with the postsynaptic cell. I'm going to have you guys draw, zoom in microscopically at a pre and a postsynaptic cell, kind of like the image there in your handout. But you actually kind of did this a little bit from handout number 2 show a simple image. You don't have to copy this one, but in your handout, draw something like what you see with figure 8.2, where there's a presynaptic cell and a postsynaptic cell. I'm going to back up, though. I want you to look really quickly and compare this to real images. On which side of the image would the presynaptic cell be? The presynaptic cell is on the right end, right? Kind of middle to the right, because I can see the what? Neurotransmitter in the vesicles. The postsynaptic cell is over here. And the same thing in the next image, it's rotated. Presynaptic cell is hands down colored red, orange. Postsynaptic cell is kind of a grayscale brown color. Our receptors are colored what in this image? Blue. Our vesicles are colored green. So draw something similar to this so you understand that there is a little bit of an interstitial space between the pre and the postsynaptic cell. Identifying all the structures that you have listed there in your handout. So axon terminal, vesicles with neurotransmitter, voltage-gated calcium channels. Where are they located? The axon terminal. Okay. We know where the cleft is. Neurotransmitter receptor is located on the which cell? The receptor is located on the postsynaptic cell. And I'll come back to the reuptake molecule in just a moment. We have not mentioned it. So drawing a pre and a postsynaptic cell. What we're noting is there's going to be the way that we can have this unidirectional communication is that there has to be a way for the presynaptic cell to release its neurotransmitter. And that's going to be the role of the voltage-gated calcium channel. So what do you think will open the voltage-gated calcium channel? The action potential that we just generated along the axon. If it reaches the terminal, and in addition to our sodium channels and our potassium channels, then calcium channels will open, and calcium will flood. Does anybody know? It actually floods 50-50. Doesn't go out, it goes into this cell. Okay, we haven't addressed its gradient yet, so good guess. Calcium, when the voltage-gated calcium channel is open, calcium floods into the cell. And what happens is it's going to trigger a series of reactions, which eventually is going to cause what? To demobilize. The vesicles are going to be mobilized. And the neurotransmitter will be, what's the process called? Do what? First exocytose so that it can diffuse. So our vesicles have our neurotransmitter. That's the release of those are regulated by the control of calcium through the gated channels, all at the presynaptic axon terminal. Yes, into the presynaptic cell. And in this image, yes, the top image. The calcium ion, the green, would actually flow into the cell. And it triggers, it's basically kind of like a second messenger system. And then the vesicles will empty their contents or exocytose the neurotransmitter. Additional questions at this point? So the neurotransmitter is not effective unless it interacts with a receptor. The receptor, go back to chapter 5, will probably open or close an ion channel, or do what? Activate a second messenger system. We're going to work on the presumption that we're open or closing an ion channel at the postsynaptic cell. What's new to us is something called a reuptake molecule and an enzyme. So what happens is, as soon as the neurotransmitter is released, it does its work. What should happen? What do you think? As soon as it does its job, we need to stop that reaction. And so the easiest way to stop it is to take an enzyme, break it down. If we break it down to something, we can actually reuse it. And so some of our presynaptic cells have what's called reuptake molecules. That, as the term implies, what do? Pick up neurotransmitter, especially if it's been recently broken down. We can take it in and do what with it? Reuse it, rebuild it, whatever. And so the reuptake molecule, oversimplified here in the far right image, right portion of the image, to basically recycle neurotransmitter is the efficient way. The enzyme in this image is immobilized down here in the membrane. Where it's located is variable. Okay? But it's just illustrating its presence. Notice that it can also bind to neurotransmitters. And so we just summarized the next bit of our information, but I'll walk back through to illustrate what happens at to cause the release of neurotransmitter. Figure 8.2 illustrates this for you. In an action, my abbreviation AP, an action potential is propagated or conducted. And notice I have a star there. The star, as we covered that in chapter 7, whether it be saltatory contiguous conduction. An action potential is conducted to the action potential. The axon terminal. That causes calcium to flow, we said, into the cell, into the presynaptic cell. What detail could we add in there? How does it get in there? Okay. By opening of our voltage gated channels. Okay. There's a series of reactions we won't address now. But what happens is calcium is going to initiate exocytosis of those vesicles, which allows for the release of neurotransmitter, and it just diffuses, collides with its receptor at the postsynaptic cell. And then what we learned is, right, point 5, we open or close gated ion channel or activate a second messenger system. I want you guys to review this real quick. I just had a call from my son's little daycare. Not good when they call. So I'm going to step out in the hall really quick. Turn to your neighbor and just y'all discuss these events, and I'll be right back. All right. So, I think we're okay there. Those 4 chemically gated channels, right? We'll keep seeing those over and over all semester long. All of these events do take a bit of time. And so it's considered to be a little bit of a delay, so maybe like 3/10 to half a second in delay between communication between the pre and the postsynaptic cell. although to you it doesn't seem very long, but it's a delay. It's not as fast as moving an ion right from one cell to another, but it's still clearly an effective way for communication. So your book addresses this, so I want to go ahead and readdress it for you guys, but y'all know that once the postsynaptic cell receives a neurotransmitter, we're going to open or close gated channels or activate second messenger systems, is what this is summarizing. Channel link, I don't know why the terminology changed, which is a chemically gated channel, or G protein, we know second messenger system is activated. So this is the terminology difference. Chemically gated channel or second messenger system are activated. Now, where I mentioned, if you remember last week, we were going to talk about what happens first, the chicken or the egg. Last week we talked about the chicken. Now we're going to talk about the egg. Like, how does a nerve cell get excited? So we're coming back to kind of making full circle. And how that happens is what's called a postsynaptic potential. It's the potential at which cell? The postsynaptic cell. It's still a cell, it still maintains resting membrane potential, but we're kind of putting it in perspective. This is telling us a cell that receives neurotransmitter from some other nerve cell. And how the postsynaptic cell responds depends upon how much neurotransmitters released and how long that neurotransmitter is bound to the receptor. And the two ways that a postsynaptic cell can respond collectively to a neurotransmitter is by either what's called excitatory or inhibitory. The terms that we've used so far is the cell will either depolarize or what? Hyperpolarize. And so if the postsynaptic cell depolarizes, it's considered to be an excitatory postsynaptic potential. But notice what I have in parentheses. What type of potential is that? It's a what? Graded. So this is a big deal. These are, yes, changes away from membrane potential, resting membrane potential, but they're what? What was the key? What makes something graded? Below threshold. Okay, so we're looking at depolarizations and hyperpolarizations, but they're changes away from that postsynaptic cell's resting membrane potential, but not yet crossing its threshold. Okay? And so an excitatory postsynaptic potential we'll call an IPSP, and we'll contrast that to an inhibitory or an IPSP, an inhibitory postsynaptic potential. So if we're to look at each in a little bit better detail, I'll come back to this image. An excitatory postsynaptic potential is a graded potential. And notice I have in italics, okay, they can initiate. They can favor an action potential, but they by themselves don't initiate. Does that make sense a little bit? The excitatory postsynaptic potentials cause the postsynaptic cell to become a little bit less negative, causing a slight depolarization, but the big thing is the postsynaptic cell doesn't cross threshold. Because it doesn't cause the postsynaptic cell, it can maybe it might be less confusing if you scratch out the word initiate and put the word favor. Does that make sense? It can favor an action potential but doesn't guarantee an action potential. Let's do that. Let's scratch out the word initiate and put favor. Is that okay? We're not guaranteeing action potential if there's an excitatory postsynaptic potential, but we're getting closer to threshold by depolarizing. And let me come back to these images as well. The whole point is to bring the postsynaptic membrane potential closer to threshold, increasing that chance for an action potential. In other words, the postsynaptic cell became a little bit more excitable. But the whole point that I was trying to make is this last bullet, the postsynaptic potential with a single excitatory postsynaptic potential is not enough to generate an action potential. So it's clearly a graded potential, but it's a depolarization getting closer to that. And so if you turn your handout to page 4, I have a plot for you, and this will go along with your homework, or your POT that's due tomorrow. On the top of the page, I want you to label this an EPSP, an excitatory postsynaptic potential. You're going to plot similarly millivoltage over time in milliseconds. You'll start a postsynaptic cell at rest, and you'll have a weak excitatory postsynaptic potential, and then make that stimulus go away, the cell will go right back to what? Resting membrane potential. You also need to indicate what? Threshold. I want you to plot a single little excitatory postsynaptic potential. The question is, how do we reference threshold? And what threshold is that minimal amount of depolarization required for a postsynaptic cell to open up voltage-gated sodium channels. Okay? So whether the value be minus 55 or minus 60 for a particular cell, if you depolarize to that voltage, we're going to guarantee an action potential. So wherever you draw it on your line, this potential should be what in respect to it? Below it.
I just didn't know what number you wanted to reference.
I tend to go with minus 55. Somewhere minus 55, minus 60 is okay. For whatever, as long as if you're differentiating a graded versus an action potential, you aren't crossing it. And the threshold is less negative than the rest. So it'll be somewhere less than the rest. But it's just your barrier, if you will, differentiate between the graded and the action potential. So if you plot your graded potential, your excitatory postsynaptic potential, I'm going to come back to these images. The postsynaptic cell would be at rest, receive some type of stimulus. It does what this upward swing is depolarizing. Stimulus goes away, then it goes right back to rest. Because what's working? The leaky channels and our gated channels are closing. The sodium-potassium pump. Remember, the pump is pumping, the leaky channels are leaking. Okay? Those are still going on. Notice there is no what? There's no hyperpolarization because we didn't activate any voltage-gated channels. So your excitatory postsynaptic potential just deviates away from rest, stimulus goes away, and goes right back to rest. But we're getting close to repetition. It'll sink in, hopefully. Everybody's image looks similar to that. Let me see what images I kind of skipped over. One image that I skipped over is illustrating, you can see the postsynaptic cell, the large cell body and the axon over there on the left-hand side. But the point of this is there's multiple presynaptic inputs that are occurring at a postsynaptic cell. If you think about the billion, remember the brain we looked at last week? Okay? Think about all the cell-to-cell interactions. Okay, so some cells may be receiving excitatory neurotransmitter at the same time as inhibitory neurotransmitter, right? So there's multiple effects. It's not just a one-to-one simple relationship. So we're going to look at, well, what one is the collective effect here? Presynaptic inputs. alternative image illustrating for you in the presynaptic cell how calcium triggers the release of our neurotransmitter. And in this case, we can see the neurotransmitter causes sodium to flow into the postsynaptic cell with what? Sodium flows in. What is the effect of sodium flows into a cell? It depolarizes. We don't know if it generates an action potential or not, but first, before it generates an action potential, it's going to have an excitatory postsynaptic potential. If it's just a little bit of sodium going in, the cell may not cross what? Threshold. So that's where we're going. Okay, if it's a minimal amount of sodium influx, just a graded potential, or we could say an excitatory postsynaptic potential. Conversely, where we really haven't looked is that not all neurotransmitters cause excitation. Some of them actually inhibit cells, which makes sense if you start to think about it in a large, like say if you go to the doctor, right, and you're going to have your wisdom teeth drawn out of your head, right? Wouldn't you want to inhibit your sensory nerves? Yeah. And so we can, and our nerve cells actually do this. You control many processes besides just pain perception. And the way they do that is what's called an inhibitory postsynaptic potential. And so the whole point of this is to prevent an action potential. It also uses our chemically gated channels. And what you'll see is probably more potassium out and or chloride ion, which is negatively charged, moving in. If you take something positive out or add something negative, the membrane becomes more negative, or we could say hyperpolarized. Purpose, or what, as I mentioned, we said we're going to have a slight hyperpolarization. We're getting further away from threshold, and at that moment in time, the probability of crossing threshold is unlikely during a small hyperpolarization. And so the membrane actually becomes less excitable. You'll notice in your handout there's a similar looking plot. So what do you think you're going to label this? IPSP, Inhibitory Postsynaptic Potential. Doing the same type of indication, plotting millivolts over time in milliseconds. Have a postsynaptic cell at rest. Give it an inhibitory neurotransmitter. Hyperpolarized neurotransmitter goes away, the cell goes back to resting membrane potential. When you do that, your plot should look something like this. And you'll notice you're just further away from threshold during that moment in time. And up till now, everything looks really pointless, right? Not generating an action potential in either scenario, right? They're not pointless. There's method behind the madness. So if you go back to that previous image, you remember where there's multiple presynaptic inputs, they can actually combine together, or the proper term is called sum. We can sum, what does that mean to sum? Add them together. I can add excitatory postsynaptic potentials with other excitatory postsynaptic potentials. It's called GRAND, another good old acronym, it's called a GRAND postsynaptic potential. The way in which our postsynaptic potentials to be summed together are by two types called temporal and spatial summation. We're going to do a hands-on scenario where a clap, not the clap, but an applause, is a graded potential, an excitatory. What could be an alternative? Let's do this will be an excitatory postsynaptic potential, and I'll differentiate between the two, and a snap will be an inhibitory. Okay? So I'm going to be a postsynaptic cell. You guys are all presynaptic cells, and you're influencing my activity. Okay? You guys are going to do spatial summation. Okay, so within your cells, choose whether you're going to be excitatory or inhibitory. Okay, so you guys just start doing whatever you want. Y'all are all excited, so go ahead and go. Okay. And so collectively, I've got a lot of excitatory input, so I can start generating active myself. Okay, does that make sense? A little bit. Lots of presynaptic inputs. You guys influence me. And y'all just illustrated what's called spatial summation. Although there were some people back there snapping, they didn't keep me from crossing threshold because all the other excitatory inputs were going on. Y'all see that? We all in tempo together? Yeah, we're doing your own thing. And you're influencing me at the same time. So that's a type of spatial summation where multiple presynaptic inputs influence a single postsynaptic cell. I might be able to do a temporal summation. Anybody want to volunteer for this one? I'm going to pick you if you don't know. No. Okay. You're going to illustrate temporal summation, and you're going to give me some great, yeah, but she's going to go regularly. You're so fast. And now I can, okay, so because she's doing this regularly, that keeps me, right? So as long as she's doing that, I'll keep doing it. So, thank you. How many inputs were during temporal summation? 1. But she was causing an excitatory postsynaptic potential what? What was she doing? Regularly, right? So, as soon as I depolarized, she sent another neurotransmitter that I can continue to cross what? Threshold. I don't know if that worked or not, but maybe an image will help a little bit. In temporal summation, the last visual that we used in class demonstration is a single presynaptic input releases neurotransmitter quickly, rapidly. And so graded potentials are generated, but before the cell can go back to what? Rest, another graded potential is added to it or summed to it, causing the cell to cross threshold. And now we have generated an action potential. Okay? So although a single graded potential doesn't seem useful, if they sum together, whether it be through temporal summation, the postsynaptic cell can cross threshold and generate an action potential. The visual help, hopefully? All right. We contrast that to spatial summation, the first scenario that we did in class. Okay, so many different presynaptic inputs are happening at the same time. We noted it wasn't just necessarily excitatory inputs, but also inhibitory inputs. If there's enough excitatory inputs, it will cause the postsynaptic cell, to cross its threshold and generate an action potential. So graded potentials seem boring until you do what with them? How? We sum them together. Okay? We sum them together by either temporal or spatial summation. These types of summations are summarized for you in the image that you have in your handout. On the far left, it's illustrating that what we're kind of agonizing going through. You're like, what is this? Single graded potential seems useless, right? If it's not summed together. If you turn to the second panel, we can see what type of summation is going on. Temporal summation, where presynaptic input releases neurotransmitter quickly so that our graded potentials stack or sum together. Now opening our voltage-gated channels and generating an action potential. Conversely, we can look at spatial summation, where two excitatory presynaptic inputs collectively sum together to generate an action potential. And the last panel is illustrating that if the excitatory and the inhibitory inputs are balanced out, it has what type of effect of a postsynaptic cell? Little to no effect. So they can actually cancel out. The postsynaptic cell still doesn't generate an action potential. So the inhibitory neurotransmitter kind of wins, if you will. What questions do you have about the purpose of summation? Or how it might happen at a postsynaptic cell? This video is going to illustrate for you what we've already walked through, so it'll hopefully put everything together. Illustrating that in the nervous system. Oh, back up. From lab. What are these little nodes? What are these little things right here? Dorsal root ganglia. Very good. The spinal cord. Action potentials are highlighted in yellow, but we know it's just ion movement. What color is the presynaptic cell? The presynaptic cell is green. Postsynaptic cell is blue. We can see the little receptors there in the postsynaptic cell. You can also see they're labeled the vesicles. We summarized today that an action potential opens up our voltage-gated calcium channels. Calcium floods in, triggering the migration of those vesicles, the exocytose neurotransmitter out into the cleft. Neurotransmitter then diffuses across the cleft, binds with receptors, and then however they're built dictates whether you're looking at an inhibitory or an excitatory postsynaptic potential. This is going to show each option, excitatory versus inhibitory. First, we're going to see an inhibitory, so which ion is going to be mobilized at the postsynaptic cell? Which ion will we see moving? An inhibitory? Well, maybe chloride, but that one's not shown. Chloride would move which way? In, but we do see potassium, so potassium's moving out. So an inhibitory postsynaptic potential illustrated in this animation. Neurotransmitter opens up, chemically gated potassium channels, and potassium flows out. So, and that postsynaptic cell just had an inhibitory postsynaptic potential. Conversely, some neurotransmitters may be what we call excitatory and allow for sodium to flood into the cell, causing us first degraded and then maybe an action potential. We also learned from figure 8.2 is once those neurotransmitters are utilized, they need to be degraded. So here we can see some extracellular fluid neurotransmitters with enzymes digesting the neurotransmitter. And here we can see in the purple the what? Reuptake molecule. And then however the process depends on which neurotransmitter it is. Good? So up till now we've been using the word neurotransmitter. We're going to throw in some specific terms. And we could talk about messenger, ligand, and now we're going to say a specific neurotransmitter, a specific receptor. At a particular synapse, the receptor and the neurotransmitter is consistent. So between the cells in my frontal lobe and my thalamus, remember we talked about how they communicate like that? They'll always have the same neurotransmitter. It's not going to have acetylcholine today and dopamine tomorrow, and then when I'm 85, something else. Okay? It's always going to have the same neurotransmitter, same receptor type of interaction. And therefore, at that same synapse, the response is going to be the same. It's always going to cause inhibition, or it's always going to cause excitation, however it's built. Okay? Some of the primary neurotransmitters that we're going to use this semester, and I don't know that I have them all listed, there may be another error in my handout. Okay? I have some. The neurotransmitters that we're going to use this semester, listed on this projection on the left, are our peripheral nervous system neurotransmitters. Okay, so I'm going to bracket these or whatever. Acetylcholine, norepinephrine, and epinephrine, you can abbreviate EPI. These peripheral neurotransmitters we're going to spend the bulk of our time with because the rest of the semester will be out of the nervous system, in the periphery. We're talking about acetylcholine, norepinephrine, and epinephrine. But they're found in the central, some of them are found in the central nervous system, along with other neurotransmitters that you've probably heard of. Heard of dopamine? Have you heard of those? Dopamine, serotonin? Central nervous system neurotransmitters. May or may not have heard of histamine, glutamate. Okay. And then you notice I have the ellipses. Other chemicals act as neurotransmitters that we wouldn't classically call neurotransmitters. So we're going to introduce these now. And the rest of the semester focus on acetylcholine and epinephrine and norepinephrine. Found this image recently, and so I'll share it with you if you would like. Looking at some of our different neurotransmitters, so going down the road, so acetylcholine, dopamine, gamma-aminobutyric acid, glutamate, glycine, norepinephrine, and serotonin. And then notice on the way that this table is laid out, that it has the way that the receptors work. They're either chemically gated sodium channels or activating what? G protein. Okay? Channel, ion channel, G protein, basically. And so you'll notice acetylcholine has two types of receptors. Dopamine only has what type of receptor? It's called a dopamine receptor, but how does it work as? Second messenger system. If you look at one called glycine, it only has chemically gated ion channels. Okay, so where we get into the difficulty in understanding how the neurotransmitter works is because the receptors may just cause depolarization or inhibition or actually activate the second messenger system. Okay, so we've got a lot of different types of receptors for different types of responses, which it makes full circle to chapter 5, where we talked about the receptor at the target cell is going to dictate the response at the target cell. And so of all the neurotransmitters that we're going to study, we can functionally classify them as either excitatory, causing what? Depolarization, or we can say our neurotransmitters are inhibitory, always causing hyperpolarization. The kick is what? They can sometimes do both based on the what? Let's just say instead of both, they can sometimes do either, depending on the receptor. The channel, the second messenger system, is it in the heart, is it in the wherever? Okay. We got a lot of diversity. It basically limits the total number of neurotransmitters that we need. So some neurotransmitters do have both excitatory or inhibitory effects at a target tissue. It's going to be. Would it make more sense if instead of using the word both, you could say either? At a particular tissue, it would be either excitation. At another tissue, it may be inhibition. At a particular tissue. And what we learned and summarized a moment ago, the receptor itself. Classic example is acetylcholine. You'll become really familiar with acetylcholine this semester. And we'll see it starting out in Unit 3 at our skeletal muscles, our somatic nervous system. What do you think it does to our skeletal muscles? It excites them, it causes the skeletal muscles to contract. Okay? However, in another muscle type, acetylcholine is actually inhibitory to the heart to cause the muscle to slow down and contract less. And so that allows your heart to rest. We'll actually see this in lab today and tomorrow. There's a cranial nerve. Does anybody know which cranial nerve slows down your heart rate? If you did the pre-lab? The vagus nerve, cranial nerve number 10, releases acetylcholine. That has an inhibitory effect at the heart. This little bit of information is a little bit out of order, and I apologize, but we've already covered this, so it will be a review. We learned that once a neurotransmitter is released, we've got to get rid of it. We said it can become inactivated. It can be taken back up into the presynaptic axon terminal, but not addressed is it may actually diffuse out of the cleft. And if it's out of the cleft, it's not exposed to what? It's out of the cleft, it's not exposed to what? Enzymes, or it is the neurotransmitter, so it's not exposed to its what? Receptors. So it's effectively ineffective if it's not in the cleft anymore. Let's look at a couple of our neurotransmitters very briefly. The first that I want to study is called acetylcholine. I acknowledged already that it's in both the central and the peripheral nervous system. We'll talk about it a lot in the peripheral nervous system, primarily in its role with skeletal muscles. The name of the receptor is a little bit tricky unless you kind of look at it a little bit closer. The receptors are called cholinergic receptors. If you look at the prefix, it says what? Choline, which is in acetylcholine. Acetylcholine binds to cholinergic receptors. Our cholinergic receptors, there are two types that either open or close ion channels or activate a second messenger system. The ones that open or close ion channels are called nicotinic receptors. The nicotinic receptors are basically gated sodium channels. The nicotinic receptors are gated sodium channels. And by default, the muscarinic must be the second messengers. And so because acetylcholine has a lot of target tissues and a lot of diversity. If we look at the nicotinic receptors in a little bit better detail, we're going to find them in three areas. One are called motor end plates. And that's if you want to put in parentheses at your skeletal muscles. Motor end plate of your skeletal muscles. And the second and third regions we'll identify better in chapter 11. I'll come back to this in just, I think it's chapter 11. So right now, just take it at face value, and I'll clarify it better when we have more time. On the ganglion cells of our sympathetic and parasympathetic nervous system. And the third area, you may sound kind of familiar, which is called the adrenal medulla. We'll come back to where these are located in chapter 11. Here's the key thing that we're focusing on right now, though. When a nicotinic receptor is bound to acetylcholine, that postsynaptic cell is going to do what? It's going to do what? Get excited, or it's going to be permeable to which ion? Sodium. It's going to depolarize. Acetylcholine bound to nicotinic receptors causes the postsynaptic cell to depolarize. Here, get excited. And here's just a simplified image of the cholinergic or nicotinic receptors, chemically gated sodium channel. Conversely, our muscarinic receptors are a lot more complex. Come back. Because it activates the second messenger system, or I've highlighted here a G protein cascade. Because it activates the second messenger system, the effect of the target cell may be inhibitory It may be excitatory. It depends on those intracellular cascading events. And I'll conclude today's discussion with an image just to help you visualize, again, the second messenger sequence. Acetylcholine binds to a muscarinic receptor, second messenger pathway, and then eventually we'll open up ion channels. In this case, what's the effect at the target cell? The end result is what? In this illustration. Inhibition. Why do you say it's inhibition? Potassium is flowing out. I'll conclude here, and then tomorrow we'll look at our other neurotransmitters, and we'll jump into Chapter 9. So be sure to read ahead, Chapter 9.